Oh, I'm Carly Jordan. I'm from the George Washington University, and I'm one of the co-PIs on BCNet. And you'll also hear from Janice Crum, who's the PI from Widener, and Cecily Bronson at Portland State University. The three of us make up the coordination team for BCNet. So we are sort of orchestrating a lot of the communication and planning for the network. Jean Woods and Liz Shea are also co-PIs for BCNet, and they're here today too. So BCNet is an NSF-supported research coordination network in undergraduate biology education, RCN UBE. And our goal is really to build a community of undergraduate educators, collections professionals, education experts, data experts that can collaborate to create opportunities to bring digitized natural history collections research into the classroom. So we are doing that by creating and supporting the implementation of course-based undergrad research experiences, or CURES, using natural history collections. And we really have the goal to expand undergrad research opportunities into populations and institutions that may have a difficult time accessing those opportunities. So we have just a few slides to define what these, how we think about cures and collections. So we are also working with CureNet for some of our training, and they define a cure as follows: that these are courses that include undergrad research experience, and that those experiences are of interest beyond the confines of the course. So the students are working on research that is new and novel, or has interest to scientists outside of the classroom, and that they are engaging in all the different processes of research. So depending on which cure they're involved with, they may be asking their own research questions. They're certainly collecting data, they're analyzing data, they're communicating with the community. And they do a lot of troubleshooting and problem solving as part of that, and build those kinds of skills, which is a really important goal for all of these cures. We know that cures are really effective ways of getting students involved in undergrad research. And there are studies that show that cures provide students with some of those same advantages as traditional research experiences. But of course, having digitized cures or having experiences in the classroom makes it a lot more accessible. So where all of the cures that BCNet is creating and supporting are using digitized natural history collections data, which you all are familiar with. We love using this kind of data because it's so accessible that students at any institution can access the same exact online data sources. Right now, it's great because these can be implemented entirely online. And so some of our funding and a big push for us this year came from trying to create experiences to replace some of the things that were lost due to shifting online due to COVID. There's so many different topics that can be used because the collections are so extensive. And some of the cures we're working on are also collaborative cures, where we have multi-institutional projects, and students at different universities across the US are contributing to a common data set for analysis. And that's also a really great opportunity. So BCNet is supporting cures by providing training, lots of training, both live and pre-recorded, to help implementers, educators who are not familiar at all with with collections, or maybe both, to learn about collections, how they can use them in the classroom, helping them understand how to navigate data portals and deal with digital data from collections. We are providing this network, sort of bringing people into the BCNet network means that they're becoming a part of a large community that contains people with expertise in lots of different fields, and we're helping them connect so that they can get the help that they need when they need it while they're implementing these cures. And we're also trying to expand out into different universities, to more diverse institutions. We have a big push to try to reach community colleges and HBCUs, Hispanic serving institutions, and places where resources might not be available for students to have traditional undergrad research experiences. So right now, this is what's happening with BCNet. So over the summer, we funded some teams of developers to create 4 natural history cures, and they are being implemented right now. We have over 30 institutions on board right now. That's probably an underestimate, actually. We just took on several more this fall, and they're spread across all different kinds of institutions. We've got over 1,500 students that are expected to participate in the cures in the 20 to 21 academic year, and they're spread across all different kinds of courses: first-year courses, upper division courses, big, large enrollment courses, small independent study courses, everything in between. We have folks that are doing this completely online, folks that are doing it in a hybrid format, synchronous and asynchronous. So the cures have proven to be really adaptable, and people are finding lots of creative ways to implement them into all different kinds of courses. So what we thought we would do now is just give you a one slide overview of each of the 4 cures that we created, so you can see what kinds of things students are doing, and then dive into one of them just a little bit more so that you can understand the structure of the cures. So Cecily is going to talk about the 4 cures.
Yeah, thanks, Carly. Okay, so the first cure that was developed was a cure for invasions. And this cure is focused mostly on first year or introductory courses where it's more instructor driven, where they will have a specific species in mind that students will test their hypotheses on. So they'll look at the historic and current distribution of this invasive species to see what characteristics that species might have that would influence its ability to disperse into native or invasive areas for that species. So they're going to use that digital natural history collections data to find the species and all the data they need for it. They might map it to look at the changes of where that species originated from and where it's going next. An example, or like all of these cures are really variable depending on how much time the course is going to dedicate to them. So a possibility with this cure is for students to just focus on one invasive species or a pair of species that could be in the same area, and one could be a native species or an invasive species, or they could both be invasive species, and to really compare those morphological characteristics or dispersal characteristics that that species might have. So an example species that one of our cures is using is the red swamp crayfish. So students are looking at that dispersal or distribution of that species and looking at it to figure out how it could have moved from its native region to an invasive region. And so our next one, this one is aimed more at advanced courses or advanced students. And this is more of a choose your own adventure. So it's how species distributions and their drivers drive the species to move. So this one, students are going to be picking their own species pairs or individual species, and they're going to be developing their own hypotheses to figure out how human activity has caused that species to move around from their native range. So they're going to compare and contrast current species distribution data using things like iNaturalist and field observations to historical data sets using the digital natural history collections. And again, this one's also super flexible and really able to be individualized by course, and it's really focused on students developing their own hypotheses and research goals, whereas the Cure for Invasions was more of an instructor-driven introduction to hypotheses and that sort of thing. So this one's more for the advanced students. So an example species that students could work with here is like the zebra mussels and how humans are moving these zebra mussels around. All right, and then this one is our morphology cure. So this one is focused on looking at the morphological characteristics of something like the purple loosestrife and how its characteristics might change based off of its native habitat to where it's invaded. And so students in this course will use the digital natural history collections, specifically herbarium images of the purple loosestrife, and they're going to be measuring specific characteristics of the purple loosestrife and comparing those measurements from the native region that purple loosestrife is from to where it's invaded and seeing if there's any differences in that. For example, this plant has caused increased fires in California where it's invaded. So they're looking at things like that. And then lastly, we have our sexual dimorphism cure, which is focused on the melanization patterns of the cabbage white butterfly, Paris rape. And they are going to be looking at the sexually selected changes of the pigmentation in the butterfly wings. And so students are again going to be looking at digital natural history collection data, specifically images of these butterflies, and determining the environmental conditions of, or the effects of environmental conditions of the pigmentation or melanization of these butterfly wings, and how sexual communication may have changed these melanization patterns. An example to expand this cure would be looking at their native range versus their introduced range and seeing how those melanization patterns may have changed. And Janice is actually going to dive into this cure more specifically, as she is currently implementing it.
Thank you, Cecily. So this is sort of a little bit of a closer look at one of the cures. And so you can see that on this slide, we have the cure development team. And so we had 3 individuals, myself, Alex Trio, who is at a PUI institution, and Alyssa, who is a graduate student at Columbia University right now. We also brought in a researcher on this species, Andrew Stower at Butler University, to work with us because he has sort of a deeper knowledge of the species and their biology. And he has served as a really strong resource for the implementers. And then you can see we have a sort of a diverse array of implementers for this cure. We had 4 of them in the fall semester and more in the spring. And you can see that this includes R1 institutions, primarily undergraduate institutions. We have some community colleges. And so we really have sort of a diverse array of institutions that this is going to be at. And we expect to have well over 800 students being involved in this cure in this academic year. So the research questions, because this is one of those 2 cures which really has it has a common data set across all of the institutions who are implementing. And so this is a big collaborative effort across these institutions. So we're interested in if traits that are sexually dimorphic in butterfly wing patterns. In this case, it's that spot on the bottom, the CU2, which is present in females and absent in males in most instances. But there is also some sexual dimorphism in the size of the M3 spot and also in the apex melanization on these wings. And so we're interested in, are they varying between the sexes more or less? And one of the reasons this is very interesting is that these melanized areas are used for thermoregulation. And so the predictions we might have for changes in wing spot may be opposite those, due to sexual selection, may be actually opposite those of what we would predict due to, thermoregulation patterns. So it's sort of tying in the 2 that's made this very interesting. Through contributing to this common data set, students at all the institutions are learning about data collection, data management and analysis. There's also a focus on scientific communication. And also, because this is across institutions, they're also getting a feeling for collaboration with the scientific community, right? With researchers that are outside their classroom. This is sort of a little bit of an overview of what one of these cures might look like as you plan it into your semester. I will say that this is one version. We have current implementers who are actually implementing this over a 15 or 16 week span. We also have some January term and spring implementers that are planning on doing it as more of a half semester or even a 4-week time period. So there is a lot of flexibility in how this would fit into your course. And so we move through different sort of trainings, starting with data portals, downloads and cleaning, moving into training them to take morphological measurements, doing some georeferencing and mapping. And you can see on the right some of the examples of some of the materials that are available through BCNet to support the implementation of these cures in the implementers' classrooms. And so there's a pretty large volume of materials available for implementers to support them, especially because a large proportion of our implementers, this is their first time working with natural history collections at all. So we invite you to get involved. We are always happy to bring in new collaborators and new implementers, whether this is something where you're interested in implementing in your course, or you'd be interested in helping someone who is an implementer already, maybe to answer questions, maybe to help with development of materials. So there's a lot of different ways to get involved and at a lot of different levels. We have our email here, which goes to actually the three presenters today, myself and Carly and Cecily. And you can also sign up for our mailing list through the website below. And I think that's it. We have a lot of people who've worked very hard on this, and this involves all of our core developers who spent a lot of time this summer working on developing all of these cures, and our collaborating developers and steering committee and coordination team who put in a lot of effort to make this doable in a very short period of time.